We would like to invite Eva already to the desk.
Thank you. First of all, thanks to the organizers for inviting me to give the talk today. So I'm Eva Cuypers. I work at the University of Maastricht, and there I'm the PI of the research group Mass Spectrometry Imaging in Perioperative Diagnostics. That includes, indeed, intraoperative diagnostics. I will not talk about that today, but also perioperative, meaning we're working towards digital pathology on the single cell level using mass spectrometry imaging, and this I will present you today. For those who don't know what mass spectrometry imaging is, first an introduction. So you basically shoot with a laser or an ion beam to ionize the compounds in your sample, which might be a tissue section, but also 2D cell lines, for instance. You shoot with a laser or an ion beam and you send the ions to the mass spectrometer where you acquire the mass spectra. But because you know also where you shoot with your laser, you have the spatial information and you can combine the mass spectrometry with the spatial information into molecular images. So these are kind of photograph kind of pictures of the compound distribution in your tissue sample. What can we acquire in the mass spectra? We can have a look at metabolites, lipids, and also proteins to some extent, so I will try to incorporate all of these today in the talk. So what's the spatial detail we are heading to at the moment? Well, you see here an overview for mass spectrometry imaging. It's a quite broad range. Why? If you work with a laser, of course it depends on the laser settings. With the laser you can go up to 5 micrometer minimum. We're already heading towards 1 micrometer, so hopefully by the end of the year we can do that too. Why is it also including 100 nanometer? The nanometer range is when you use an ion beam, so that's SIMS imaging. I will not include that in my talk today. We also do that in the lab, also on single cell level, but I will not include this data today. So I mainly will focus on the MALDI using the laser, the MALDI mass spectrometry imaging today. So how detailed can we go? This is just to give you an example. What do you see here? These are molecular images of 2D cell lines which are grown on an ITO slide. Bigger cells like fibroblasts, but also the tiny ones like the cardiomyocytes. There is just one compound, or in the case of macrophages, 2 compounds selected, mostly lipids, to show you how these cells look like. Even in the macrophages, depending on how big the cells are, you can even make a distinction between the inner core of the cell and, for instance, the tails. You see the difference between the red and the green compound in this case. So these images we can acquire routinely at the moment already. You need 2D cell cultures which you grow on a certain way on an ITO slide, which is coated with polylysine, and then you let them grow overnight, and you can immediately image these cells on the ITO slides. So we started to make databases, single-cell databases, based on these cell images, actually. So what did we do? We took 14 different breast cancer cell lines, and we grow them on different cells, on different slides, and we imaged them all. This is just an example. We did a lot of more slides than this in order to get also to check the reproducibility. You see all different cell lines. They look kind of differently. Of course, also the mass spec data are different. I didn't include the mass spec data, but here you have an example of a mass spectrum of one single cell. So we took the image, the molecular spectrum, the mass spectrum of every single cell, and put that into a recognition system. What did we include? We measured mostly between 100 and 1,000 Daltons, so it's mainly metabolites and lipids. Of course, lipids are the most abundant ones. Metabolites are also, we know that in the recognition model, but are not a very significant factor in the recognition model. So we built a recognition model, in this case for breast cancer cell lines, 14 different breast cancer cell lines. Of course, now we wanted to know, can we, based on this recognition model, recognize these cell types also in a patient tissue or in a xenograft tissue? So this is what we basically did. We had breast cancer xenograft from mice. On the left, you see the offline recognition, meaning we had a section and we imaged it, and after the image, we did a cell recognition based on the recognition model, and it identified where the specific cell types for that xenograft were. On the right, you see a real-life video. It's not speeded up, What you see in here is you see the laser shooting. This is the H&E staining to give you an idea because there is a rupture in the tissue. When the laser is on the rupture of the tissue, you get an outlier, which is normal because there are no cells. But when every spot the laser is hitting, you get the identification of that specific cell type based on the recognition we made, on the recognition model we made from the 2D cell lines. So we can do this offline and also online. Online might be important when you want to do that intraoperatively immediately during the surgery, for instance. It can go quite fast. Of course, if you want to translate into clinics, in most cases, the pathologists are not always interested in the cell types, but more into the prognostic or the therapy response markers. And for breast cancer, that's mainly the oestrogen post-progesterone receptor state. So we built basically a new model based on that state, as you can see here on the right. And we did again on a xenograft, a mice xenograft, the recognition of the receptor state. So also that is possible, all based on the 2D imaging mass spectrometry data and the mass spectra that we extracted from these cells. And then my colleague, Brit Klaas, she played around a bit with the oxygen levels in these 2D cell cultures, and she grew in normoxia conditions, 1% oxygen, 0.2% oxygen, and then anoxia conditions, the different cell types. Why? Well, one cell type, but in the different conditions. Why? Because it's important also in pathology. What you can see here on the right, it's a CA9 staining, which is typically for hypoxia conditions, and that's what pathologists mostly are interested in. So we basically made, again, the same model, but then of the breast cancer cells grown in different oxygen conditions. And what you can see here is that, for instance, where there is dark brown, which is a hypoxic region, it's also indicated here in red and at some part in blue. So it matches exactly what the pathologist stated as this is a hypoxia region. So I briefly showed you what is possible with mass spectrometry imaging, mainly with MALDI. So we used the MALDI laser in these 2D cell lines. We use this mass spectra on the 2D cell lines to use artificial intelligence, what they call it with a nice word, to build recognition models and then go towards digital pathology, offline or online. What I showed you so far is mainly on metabolites and lipids, what I showed you in the spectra between 100 and 1,000 Dalton. But can we also do proteomics on the single cell level? That's a very often occurring question we get. So my colleague, Andre, put really a lot of time and effort in trying to do proteomics on these 2D cell cultures for breast cancer. And very recently, he managed to have nice images of these breast cancer cell lines in 2D after androgen retrieval and trypsin digestion. So what you see on the right, it's a spectrum of one single cell after trypsin digestion. So it means these cells are about 15, 20 micrometers, so you have less than 10 spectra per cell. But even though he was able to find some peptides after the trypsin digestion. So we're heading towards single-cell proteomics at the moment. So what I showed you so far is that we start from 2D cell lines, from single-cell profiles, and we use these profiles to build recognition models for in-tissue recognition and digital pathology. But there is quite a big challenge. Not all cells are available in 2D cell lines, and also there is an influence, so we didn't have like 100% accuracy on the recognition models. Of course, because in-tissue environment is still something else. There is cell-cell communication, there is interaction, so we can improve the models, we believe. And the only way to do that is to start from the tissue sections themselves and then try to find where or which cell types and use this information to put in the databases. So basically, rather than start from a 2D cell line, we start from the tissue itself, find out where are which cell lines, and then we use these molecular markers to put in the database and the recognition model. It sounds easy, but it's not. In practice, it's not so easy. So what you see here is a rat with a glioblastoma, a brain sample, and on the left you see the MSI result. This is just to show you a lipid. You clearly can see the brain tumor. And after the MSI on the same tissue section, we do staining for all kinds of cells. We can today, in best case, go up to 10 to 14 markers staining after MSI. Of course, then you have the molecular spectra, you have the fluorescent spectra, so you know, for instance, where are the astrocytes. If you can do an overlay, you can extract the information from the astrocytes, for instance, from both images. We're more interested in human glioblastoma samples, so this is what you can see on the left is an MSI, so a mass spec imaging from a human glioblastoma sample. We first did the MSI, and afterwards we did the staining for macrophages, because macrophages are thought to be a key player in the evolution of these glioblastomas. So the brown areas are basically the macrophage stainings, the blue ones are the nuclei staining to know where are the other cells. Then we do an overlay, and based on this overlay, we have an in-house written program where we can say, rank my cells, for instance, all the highest brown stained cells. So these are the macrophages, and compare them to the lowest stained cells, which are the other cells in this case, and find the differences. So you can see here that we find differences, specifically mostly lipid masses, which are indicated as these are interesting in the macrophages or higher in abundance in the macrophages. And when we then go back to the images from the MSI, you see that the areas from these masses are indeed more or less showing the same distribution as where we saw the brown staining from the macrophages. It sounds very easy again in theory and also in practice. What's the biggest challenge in this overlaying strategy? That's the exactness of the overlaying, because we measure all this MSI data on 5 micrometers, so meaning that every 5 micrometer we have a mass spectrum and molecular information. So that means also that our overlay of the staining with the MSI needs to be smaller than 5 micrometer, which is hardly impossible to do by eye. So software is, again, also we heard it in the previous talk, software is really an important part when we can certainly improve. How did we optimize or try to do it the best we can at the moment? At the moment, in the glass slide, where we put the tissue on afterwards, the tissue section, we engrave with a laser different signs, and we fill these little gaps with mass and a fluorescent stained marker. So we can see the mass in the MSI results, and we can see the fluorescence from the same slide on the fluorescence imaging. The thickness of this is here about 6 micrometer. We can go even lower. Of course, if we go too low, we cannot see it anymore in the MSI. But this helps us improving a lot the overlay error that we, of course, want to limit as much as possible, because otherwise we annotate a different or the wrong mass spectrum to the wrong cell type. So this is really an important parameter if we want to build decent and reliable databases. There is one step in between, which we're also working on, between the pure, what we call cell types, and then the patient tissue samples, which is the patient-derived cell lines. These are patient-derived cell lines, again, from glioblastoma samples, so we have the tissue, but also the patient-derived cell lines from the same patients. This came basically from a request from a collaborator. Why? Because they see differences between the primary glioblastoma cell lines and the recurrent glioblastoma cell lines from the same patient. They see that they react differently on all kinds of treatments. They cannot find any difference in the genomics data, not in the transcriptomics data, but there is something happening because the recurrent patient-derived cell lines do not react anymore to the therapy. So we are currently investigating what is happening there. Are there cell types changing? Is there something changing in metabolites or lipids? So what you can see on the left here is the patient-derived cell line mass spec imaging. You already see that there are kind of different cell types in this, in this image. But what you can also see is that there are different cell types, of course, because it's a patient-derived cell lines, it's not one pure cell type. So there are astrocytes, macrophages, and so on in the same. So we can basically also overlay here based on fluorescent staining where or which cell types and what's happening with these cell types. All of this to go to what's the final goal for us is going to molecular pathology and digital pathology. So this is an overview of what we can do at the moment. This is from my colleague who already described and published that we can indicate based on stainings where are the different cell types, and then on these cell types where are which compounds. But we can also even go further based on the cellular databases, where are the tumor cells, the necrotic cells, the normal areas, and even within the tumor region we can already identify some cell types in the tumor region by mass spectrometry imaging based on the cellular databases. Then, to give you an overview, because it all sounds really nice, and there are a lot of pros on the single-cell MSI workflow. We can do 5 micrometer MSI images. We can define cell types, hypoxic states, hormone receptor states, and we can use the staining and the overlay to validate pathologically wise the results we have from our recognition models, and we can even expand our cell databases. But there are, of course, also a few challenges at the moment. Since we work on 5 micrometer, if you want to do an MSMS validation of your, for instance, your lipid identity, it's very hard to do on 5 micrometer because the abundances of the peaks are very low. So at the moment, we do it on a bigger spot size, like 20 micrometer. Then we have abundances high enough to do MSMS. But of course, then if you have small cells, it's not single cell anymore. Then there is also, I showed you already, we can do lipidomics, metabolomics, proteomics, but in many cases we have to choose because we have other run methods for metabolites and for lipids in order to increase the abundances of the compounds. And if you want to do proteomics, you need a trypsin digestion, so it's impossible to do on the same cells, on the same slide, on the same section of your sample. So we wanted to, and this is to wrap up, we wanted to see how much can we do with one single tissue section, how much information can we get out of it with MSI, and that in combination with an extraction and LCMS. So what we standardly do now is we spray a matrix, also including an internal standard in order to, this internal standard is a lipid splash mix, which we spray together with the standard, and then we do an MSI to extract the metabolites and also the lipids, and we try to also quantify the lipids wherever it's possible. Then based on these images from mass spec imaging, we do an automatic segmentation, and this script is automatically translated to the laser microdissection microscope, where we can burn certain areas and extract from these areas the lipids and the proteins with LCMS. So how does it look like? On the left here you see just images and with different regions. So also this is a human glioblastoma sample. You have the tumor regions, you have the hypoxic regions, and then you have the necrotic regions. Here again the CA9 staining to show you where the hypoxic region is and the necrotic region. This segmentation is done unsupervised, but it matches quite nicely with the masses that we found here specific for the different regions. And this is how the LMD protocol looks like. So you basically cut out this region, in this case the necrotic region, and then do an extraction for lipidomics and proteomics on the same sample. To show you, these are very new results, so they still need to be confirmed, but we can find, based on this workflow, even though we did MSI before on the same sample, we still can find very interesting proteins and protein pathways that are upregulated in the tumor. So this is, at the moment, the most information we can get out from one single tumor section, and let's see where we can head towards within a few years. So in conclusion, I hope I could show you that single-cell MSI is possible for metabolomics, lipidomics, and proteomics. We can use these MSI profiles to build recognition models, which we can use to recognize different cell types in a tissue environment. We can also use the immunohistochemistry staining to pathologically validate the results we get from our recognition models, and we can expand our databases. And at the end, we can also combine the MSI on the same tissue sample and do lipidomics and proteomics based on the extraction. Of course, I want also to thank all the collaborators and all the sponsors, more specifically Tim, Brit, and Andre, who also did a large part of the work that I was able to present today. And I want to thank you for your attention.